In 2012, it was reported that approximately 17 million American adults abuse alcohol. As a prevalent health concern in the United States, it isn't surprising that the neurobiological and psychological processes that regulate alcoholism are heavily studied and well known in our society. However, an oft-forgotten perspective of alcohol abuse is its evolutionary roots, which date back to our primate ancestors. Alcohol tolerance is not equally distributed throughout the world's population. In fact, it is often genetically predisposed how well a person can metabolize alcohol by how active the enzyme alcohol dehydrogenase, also known as ADH4, facilitates the conversion between consumed alcoholic beverages to aldehydes or ketones. Scientists determined this by reading the DNA responsible for ADH4 in 27 modern primate species and performed genetic mapping on a primate family tree in order to find the origin of the formation of this enzyme. Their research showed, in fact, that most primates would not have been able to digest alcohol until about 10 million years ago. ADH4 works 50 times more efficiently than its ancestral enzyme, and this could describe the evolution of alcoholic drinking tendencies in the primates studied since they started to consume much more alcohol in later generations. These ancient ape ancestors roamed the earth when its surface began cooling down, and the primates gradually spent more time on the ground, beginning to consume fermented ethanol-containing fruits. Apes that could digest this ethanol survived longer than ones that could not, so this enzyme was positively selected to remain in the population.
The history of our infatuation with alcohol has prehistoric roots in the fruit-gathering nature of our ancestors. Hunting for fruit high up in the canopy of the tropical rainforest is a daunting task, given that the sea of branches and broad, expansive leaves greatly limits the range of vision of fruit-foraging primates. Even the vibrant visual cues of ripening fruit are oftentimes not enough, driving these creatures to rely on olfactory acuity. Aromatized molecules, whose production is driven by the mutualistic relationship of flower-bearing plants and fruit-foraging creatures, are the reason why we are able to discern the difference between an orange or a lime or a coconut based on their smell alone. However, there is an aromatized molecule that is common to all of these fruits, and just about every fruit in existence for that matter: alcohol. Wherever there is sugar and yeast, there is alcohol, and our fruit-binging forebears knew this. In fact, the alcohol content of a fruit is also a fairly reliable indicator of the sugar content, and thus the nutritional value of a fruit. Beyond its ability to tell a primate that there is food nearby, alcohol's appetite-stimulating effects have been demonstrated in humans and in other mammals as well. It's possible that alcohol may have driven primates to not only eat more, but to eat quicker, limiting the time spent vulnerably gorging on fruit.
Essentially, the hypothesis explaining why human brains link reward-motivated pathways with alcohol consumption is because ethanol was once associated with a food source in our ancestors 10 million years ago. At the time, the Earth's surface started to cool down. Primates gradually spent more time on the ground and started to consume fermented fruits, which consist of ethanol. Based on the concept of natural selection theory, negative selection weeded out individuals that couldn't fit. About 10,000 years ago, alcohol was intentionally produced by fermenting fruits for special occasions and religious events. Nowadays, the sense of drinking in moderation has been skewed and causing numerous health consequences such as fatal cirrhosis or ulcers within the digestive system, and many alcohol-associated cancers. Alcoholics may experience neurological damages that lead to a sharp decrease in judgment, decision-making, and cognitive skills. Finally, family and personal history can be used to predict the risk and study genetic medicines for alcohol dependence.